Hello, welcome to today's webinar. My name is Catherine Bryant and I'm the founder of the Speak Foundation. Today we have an exciting webinar presented to the LGMD2I community from ML Biosolutions. The webinar is on a promising new oral treatment for LGMD2I, an update on the development of BBP-418, otherwise known as Ribitol. I will be introducing our speakers, Dr. Sproule and Dr. Johnson, in just a moment. And we also are excited to let you know that this webinar from ML Biosolutions, to let you know a little bit about this company, they were founded by a family whose child was diagnosed with LGMD2I. ML Biosolutions is a member of the Bridge Bio family, a team of experienced drug discoverers, developers, and innovators working to create life-altering medicines that target genetic diseases. So this webinar is going to be helpful to the 2I community because this is an exciting new potential treatment for the 2I community. Now I'm going to introduce our speakers who are here with us today. We have Dr. Nick Johnson. Dr. Nick Johnson is an associate professor of neurology and human and molecular genetics and vice chair of research in neurology at Virginia Commonwealth University, with a focus in inherited neuromuscular disorders. He received his undergraduate degree in molecular and cellular biology and psychology at the University of Arizona. We also have with us Dr. Douglas Sproule. He is the Chief Medical Officer at ML Biosolutions. Doug earned his MD from Columbia University College of Physicians and Surgeons, a Master's of Science in Biostats from Columbia University, and an AB in Biochemical Sciences from Harvard University. I want to welcome these gentlemen to be with us today, and you can start sharing your slide deck. And remember, everyone, there will be a Q&A after their presentation, so make sure to listen because we'll probably answer lots of questions you have. Gentlemen, go ahead and share.
Thank you for giving us the opportunity to meet with the community and talk a little bit about our program as well as the disease itself. I'm going to talk briefly, and we'll pass the baton back and forth to the expert in the room, my esteemed colleague Johnson. I'm going to talk about our company. So ML Biosolutions is a small company that was created out of the work in a laboratory at Atrium Health in North Carolina. It is part of a consortium of companies under under a company called Bridge Bio, which is the parent company for ML Biosolutions. This company was created to try to create a new pathway to enhance the pace and success of early clinical development programs and to bridge a major challenge that we have in early drug development, which is the lack of resources and the lack of expertise, particularly on the pharmaceutical development side, that often limits programs. One of the great aspects of working in a company like Bridge Bio is the fact that it does have immense resources as far as expertise in a number of areas that we're able to leverage at ML Bio to help us advance the clinical program as fast as we can, which is our critical goal. Every minute counts. It counts for you, it counts for us, it counts for everybody involved who's suffering from this disease. And I'm not going to talk through this, but just to kind of give people a flavor, this is a company that has a diverse portfolio of drug products spanning the gamut for a large number of rare diseases. And rare diseases are obviously a major focus for our company as well as many others. But ML Bio is specifically and laser focused on LGMD2I. This is the basic core group. The Chief Executive Officer is a gentleman by the name of George McClendon. My Chief Scientific Officer colleague is a woman named Uma Sinha. There's me, there's Nick. We have a number of other consultants who provide expertise in a number of different areas, as well as a broad outreach to the clinical community. I wanted to talk a little bit about drug development. in general and how it specifically applies to our program in LGMD2I to give a little bit of a flavor and understanding of the complexity, the challenges, and frankly why things take the time that they take, which I don't feel is really well understood, appreciated. Certainly before joining industry, it was foreign to me as well. The first stage in development of any drug is what's called the preclinical phase, and this is a period of time where you're testing a drug on animals, often mice, pigs, maybe dogs, non-human primates. It really depends on the program, the specifics of the product. And what you're looking to do is to demonstrate basic safety, the limits of how much of a drug you potentially give. You're also trying, if you have the opportunity, to test in a model animal of the disease, does this drug actually work? Once you've established some reasonable bounds of what could be a safe and effective dose in the animals, you then would proceed to what's called phase 1 testing. And phase 1 testing is often, though not always, performed in healthy volunteers, so individuals who are not affected by the disease in question. What you're typically looking for is to understand the safety of the drug, what doses could be administered in a safe fashion to actual human patients, and so this type of study is typically performed in set laboratories, again using healthy volunteers, and it's something that ideally is something that is performed relatively quickly and rapidly. Assuming you do demonstrate that there is a safe and reasonably safe drug dose that you could administer, you then would proceed to what's called phase 2. And phase 2 are typically smaller studies in a relatively modest number of patients who are affected by the disease. And the goal of this is to establish that this drug is safe and administered to actual human patients affected by the disease, but also to show some signs of efficacy, that you have a dose that's a reasonable dose that you could actually expect would work and actually benefit patients when provided in the real world. And assuming you are successful there, you would proceed to what's called phase 3. Phase 3 is typically a larger study performed in a larger group of patients, often controlled, meaning that some patients would receive the drug and others would receive a dummy drug, a placebo. And the idea and the goal of this phase of development, this is what's called the pivotal study, is to demonstrate that there's a clinically meaningful benefit of patients receiving this drug for their disease. They receive the drug and they have some actual meaningful benefit on their lives from receiving it versus not. And that's what will typically go to be the primary resource for the pursuit of regulatory approval and availability in the marketplace. There's also what's often called phase 4, which are typically very large studies that are performed in what's called a post-marketing phase, so after a drug is actually available, to establish the long-term efficacy, long-term safety, long-term benefits, and risks for patients. In rare disease, what that typically manifests in is what's called a disease registry, where patients would be followed with a given disease on and off therapy in a way to really kind of see what happens in the long term. So when you're thinking specifically to ML Bio and our product, a product called BBP-418, commonly known as Ribitol, is an ongoing effort to try to, as fast as we can, move through these stages. We've done a lot of work in our preclinical phase of development, so there's still ongoing preclinical chronic toxicology studies where we see how long it takes, or see the safety in animals that receive the drug over long periods of time, and that's something that's wrapping up hopefully over the course of this year and early next. We're midway in our phase 1 study. We'll hopefully be completing in the next couple months, that will establish the safe range of dosing that we could administer to human patients. This was something that we've been able to complete despite the COVID pandemic, which obviously has made it very challenging to do any sort of clinical research. And we're very excited to embark in the early part of next year on our phase 2 study, which will be a small study of a small number of patients who with LGMD2I who will receive BBP-418 in the context of a small pilot open label study. And this is going to be a very important study for us. It'll be the first experience that we have seeing how this drug might work in actual patients affected with the disease, and will help us establish the dose range that we would think would be effective in patients affected by this disease. We are, depending on the success and the readout from that study, we will be hopefully expeditiously and as quickly as possible moving into a large phase 3 study, which would be our pivotal study that would hopefully demonstrate the clinical effectiveness of this drug and its impact on patients, but would serve as the cornerstone and the critical data that would support our conversations with regulatory authorities. And this study will also, as Nick will talk about a little more, enroll patients ideally directly from the ongoing Lead-in Study, Lead-in non-interventional study that is currently ongoing in a number of sites. And this will hopefully create the core population of patients who will be able to enroll in that study. So I'm going to talk briefly about how Ribitol works, or BBP-418, otherwise known as Ribitol works. This is a drug that is actually a natural product. In the normal state of affairs, when you're not affected by LGMD2I, there's a process called glycosylation that occurs on a protein called alpha-dystroglycan. Simplistically, what glycosylation is a placement of these sugars onto that protein to allow it to work effectively. So what happens is ribitol phosphate gets converted into something called ribitol CDP ribitol. Then the FKRP gene, which is the gene that is aberrant in LGMD2I, attaches that sugar to alpha-dystroglycan. And that allows the alpha-dystroglycan protein to work properly. What does it do? Simplistically, obviously there's always an additional complexity. It kind of serves as a shock absorber for the muscle and muscle cells as when you contract and relax your muscles. When FKRP doesn't work correctly, you don't have effective glycosylation of that protein, so it doesn't work as effectively. So that shock absorber is not working as well as it should. This leads to injury to the muscle, leads to cell death, turnover, fibrosis, loss of muscle mass, and all the things that ultimately manifest as the clinical disease that you get that patients affected by LGMD2I know and appreciate. So what Ribitol, what BBP-418 is meant to do is to basically provide a huge amount of substrate. So an excess amount of that BBP-418 causes there to be a large amount of CDP ribitol, and that will hopefully increase the amount that is attached successfully to that alpha-dystroglycan protein, causing it to work better and slow down, or ideally potentially even arrest some of that progression that we ultimately see in muscle loss and injury. The first reaction I had when I joined the program was a little bit of an eye roll that this would actually work, but the preclinical data from the animals is extremely impressive. So on the left, you see what the muscle should normally look like with a stain called a metroglycan stain. You see what happens when it's untreated; it's basically non-existent, and there's a significant increase in staining, meaning a significant increase in expression of glycosylated alpha-dystroglycan after administration of BBP-418 to the mice. And so what you're able to go from is very low levels to something approaching 50% of what you see in an unaffected wild-type mouse. So this also correlates with a lot of impact on clinical measures. And so it's really really an exciting observation that certainly would make it creates us a lot of enthusiasm from our perspective, and the prospect that this might be beneficial in patients as well. So I'm going to pass the baton to Nick to talk a little bit more about the Lead-in Study and a little bit more about the disease as well.
Yeah, so I think one of the real challenges as Doug alluded to, is how do we tell whether or not BBP-418 is going to be effective? I think nobody on this call wants people to take a medication that's either not safe for them to take, or spend their money on a medicine that's not going to change the outcome of their disease. And so in order to do that, it's really critical that we work through how to design those clinical trials in such a way that they show that the medicine is going to be both safe and effective. And the challenge in LGMD2I is that the prior natural history studies really don't capture the patient population that would be a benefit, the full patient population that would benefit from a medication like BBP-418. And may not have been conducted over the period of time of which we would expect a clinical trial to be conducted. So ML Bio has partnered with the GRASP LGMD Consortium to conduct an additional natural history or lead-in study for the LGMD2I. Allows us to collect information about the endpoints and other tools to reflect changes in these patients. So these types of studies are really important for developing effective treatments for the disease. If you want to participate in the Lead-in Study, we really do want you to, then you should contact Brittany Holmberg. Her phone number and email address are down there underneath. Next slide. So this study so far has 30 participants in it. We're hoping for 80, so there's still lots of room. If you have limb-girdle muscular dystrophy 2I, and I would be willing to participate. There are 7 sites that are currently active. You can see them. My own site of Virginia Commonwealth here in Richmond, but also University of Iowa, Kansas University Medical Center, University of California in Irvine, University of Colorado, Kennedy Krieger in Baltimore, and then Atrium Health in Charlotte, North Carolina. We will have additional sites that will open over time. Those include Dr. Bissing's site in Copenhagen, the Nationwide Children's Hospital in Columbus, Ohio, and then Brigham and Women's Hospital in Boston, Massachusetts, and last but not least, Washington University in St. Louis, Missouri. And so, we think that, and one more thing to say about this study is that because of changes associated with COVID-19, we've allowed for remote enrollment and assessment, so you can participate in the study just as you're listening to us now. Next slide. The important thing to remember is that this is really an exciting time in limb-girdle muscular dystrophy. Therapies such as BBP-418, which is Ribitol, have a fairly unique mechanism of action and a lot of advantages because it's likely that you'd be able to take this orally and that the safety profile is likely quite good. But there's other clinical development programs that are underway, most specifically relating to gene replacement therapy. And so these types of programs take the gene that's missing. If you go back to the slide that Doug had early on, you could see that the FKRP was missing. These types of therapies take the missing protein and missing gene, and then use a virus to deliver it back into the muscle cells. So many of these programs are still being tested for safety in animal models and efficacy, so not quite ready for prime time yet, but certainly other things coming down the pipe at the same time. Next slide. So, while we're on the topic of what to do about limb-girdle muscular dystrophy, I want to take a moment just to make sure we're all on the same page about what is LGMD2I. So this is an autosomal recessive form of limb-girdle muscular dystrophy. As Doug mentioned, it's the result of a gene mutation from loss of a gene called FKRP. And in order to get this disease, you have to have one bad copy from each parent. So if you have just one bad copy, that makes you a carrier for the underlying mutation, but to the best of our knowledge, not somebody who has symptoms of the disease. One of the unique things about limb-girdle 2I compared to some of the other limb-girdles is that there's a strong founder mutation. So there's a very common mutation. You can see it there, 826C to an A, or 276I is the other way people will refer to it. And this is really referred to as, most people have a mutation, have both genes have a mutation in this particular point. And that's likely from a Northern European ancestral founder effect. Typically, people with a homozygous mutation tend to have a slower and less severe disease course. I do not want anybody who's listening to this to think that that means that they don't have really serious disease and that it doesn't significantly affect their quality of life. Other patients will often have a copy of their common mutation and any other gene mutation, and these are typically referred to as heterozygotes and tend to have a more rapidly progressive severe course. Next slide, please. Just to make it really confusing for you, the naming system for how we name limb-girdle muscular dystrophy has changed. It doesn't mean the disease has changed really at all, but in the past, if it was autosomal recessive, which means you have to have two bad copies, it was denoted by a 2 versus a 1, and that I at the end indicates which gene was mutated. But unfortunately, there have been a number of new forms of limb-girdle muscular dystrophy that were described, and so they got to the end of the alphabet in terms of the number of genes that might cause limb-girdle muscular dystrophy. So they've switched the naming system around. It's now called LGMD-R9. So R is in recessive and 9 is in the 9th form of limb-girdle muscular dystrophy, FKRP related. But that's basically, it's the same disease, but you might hear people talk about it as R9 or 2I. It's just the same thing, just new or old system. As Doug said, this is really a disorder where the alpha-dystroglycan is missing a sugar that gets attached to it, and so that's what that BBP-418 helps ameliorate. There's been really exciting work, as Doug mentioned, about how that BBP-418 acts through that system. Next slide, please. So what can you do about it? Well, limb-girdle muscular dystrophy, as you're aware, causes progressive weakness starting really of the hip and shoulders. And so there's a couple things. Number one, it's really important to get that appropriate genetic diagnosis. One of the things that's really revolutionized our clinical care of these patients is the ability to get that genetic testing at free or quite reduced rates. That helps us understand whether or not there's going to be heart or lung problems associated with that type of subtype. But more importantly to you and to me is that because these new genetic or precision-based therapies, it lets us know whether or not you're going to be eligible for this. So only people that to date that we think are going to be benefit from something like Ribitol or 418 would be somebody with a mutation in the alpha-dystroglycan pathway, not somebody with LGMD2I, for example. So you do want to have that multidisciplinary care through breathing, through an MDA clinic. The GRASS LGMD consortium is formed by clinicians who have a lot of care and expertise in taking care of patients with limb-girdle muscular dystrophy. And seeing one of those sites in particular will, they have multidisciplinary teams that can help monitor for cardiac or breathing problems, but also help with PT and OT, mobility devices as needed, and then of course psychosocial and family support. Next slide. So as I alluded to on the last slide, this is a really big deal. It has revolutionized our care of you and patients with limb-girdle muscular dystrophy. There are 2 free programs that you can use to get free genetic testing. One's the Lantern Project. It's offered through PerkinElmer. The other is detect muscular dystrophy that's offered through Invitae. The services that you can see there are provided, but really both get you to the same place. They'll get you a genetic test that can confirm the subtype that you have. Next slide. And last but not least, I'm here encouraging you to work with us as a partnership. We can come up with all the coolest, fanciest types of drugs that might help you with your disease, but unless we work with you to test whether or not those are effective, it's not going anywhere, just really well-treated mice. So this is a partnership that we engage as clinical investigators with you as an individual with limb-girdle muscular dystrophy 2I. And we need your help. We need you to participate. We need that, these trials, you can see on this slide there, tell us a lot of things. They, number one, tell us how that disease progresses. They help us better counsel you about what to expect and how best to mitigate some of the complications of your condition. They help us understand the mechanism of the disease and potentially discover new treatment targets. And then most importantly, our ability to measure change quantitatively. So not just saying, well, I know I got a little bit worse next year, but really, we think that your walking got about 5% worse. That's the kind of information that we need to design drug trials so that they can be effective in showing whether or not that therapy is going to be beneficial to you. We do take care, both as part of the Grassroots LGMD Consortium, and I know through ML Biosolutions, to engage you as patients during the trial design process to make sure that the burdens that are placed are as minimal as possible. Next slide. And so that wraps it up, and we can open up for questions. But really just a friendly reminder to contact Brittany Holmberg, which you can see there, email and phone number, if you have any questions about that lead-in study or helping participate. We'd have plenty of slots left.
Great. Thank you guys so much. You did a great job. We have some questions from the audience, and I know these are exciting times for everyone with 2I, which that is the form of limb-girdle I have as well. So this is exciting for me. Here's our first question. If I receive BBP-418 as part of the clinical trial, would I still be eligible to receive a gene therapy in the future for 2I?
Yes, so I'll take it from the standpoint of our company, and then I'll let Nick kind of talk about it in a general sense. I think every company and every clinical trial will have its own entry criteria, and there are exclusions that different studies would have. At this point, my expectation is that either one of two things would happen from the results of our clinical trial. One, the one I hope for, and we all hope for, is that this demonstrates that these studies demonstrate that this drug is safe and effective, and it becomes available in the standard of care for the disease. The other is that it fails that, and it's demonstrated to not work, in which case it would be not be something that people would be recommended to take. And so from my perspective, in either one of those circumstances, there presumably would be an opportunity for patients to participate in future drug trials. But again, that's entirely speculative, and it's going to be decided by certainly different people than me. It would presumably involve people like Nick and other of his colleagues.
Yeah, I think that question is very challenging to answer because if it's approved in clinical care, it's likely, as Doug said, to be included in those clinical trials. How those trials are designed, we don't know exactly, but generally speaking, there is some wiggle room in terms of how they're designed and making sure that patients are accommodated. So I wouldn't let the idea that there might be future gene therapies prevent you from getting help, potential help with your treatment now.
Oh, thank you. We have also another really good question. Is there a minimum age to participate in the trial?
So the lead-in study, the minimum age right now is 10. Doug can talk a little bit more about developing therapies in children, but for right now it's 10.
Yeah, and our studies are going to mirror that. we'll have to work out whether it's 10 or 12. It's going to be in that vicinity. We will be exploring additional animal studies that would allow us to push that age range down even further. But that will be a little bit slower to develop. Our ultimate goal is to develop this product in a way that it would be available for any child or adult affected by this disease.
Okay, great. What are your thoughts about over-the-counter D-ribose? Is it the same as BBP-418? And if I'm taking it, should I stop it in order to participate in the trial?
So I will speak clinically first, and then Doug can chime in terms of the clinical trial design. So D-ribose, when taken, actually competes with glucose. So what your body needs to maintain its energy and for you to stay alive. And so I would encourage lots of caution when you're thinking about taking D-ribose over the counter, because taking a particularly high doses, you may cause yourself to get hypoglycemia, not feel very well, perhaps even faint with that. So from a clinical standpoint, I would encourage people to not use high doses of D-ribose because of that safety concern. Just so you're clear, so we're all clear, Ribitol as a precursor doesn't have that same safety concern. And then do you want to talk about the clinical trial, Doug?
Yeah, from a clinical trial perspective, obviously things are in development. One thing I can pretty much take to the bank is that patients will be required to wash out and stop that supplement before participating in clinical trials.
Okay. How will you know if BBP-418 is working? What assessments are you going to use?
So, I think we turn back to people with LGMD to think about what affects them the most. And so we're going to be looking at how people walk, how they move around. If you're not walking, how your upper extremity is working. And generally speaking, as a composite, how does it all put together? So
that, and then obviously there's additional aspects, including what's called patient-reported outcomes. So how does it affect other aspects of your life? And the ultimate goal and the mandate and the obligation with the regulatory authorities is that we need to demonstrate that there's a clinically meaningful impact on patients' lives. So that's where impacts on function, on walking, and then on quality of life are critical measures.
Okay, that's good. If I'm in the lead-in study, will I automatically enroll in the clinical trial?
So I'm going to clarify the term automatic, because automatic implies that it just happens without having to do anything. Our anticipation, our expectation, and our personal obligation, personal is the right term, but our obligation as a company to the participants of the lead-in study is that we're going to do everything possible to allow enrollment of every patient who participates in the lead-in study, provided that it's safe to do so. All right, so we will have entry criteria that are mostly around obviously needing to have the disease, which is part of being in the lead-in study, but also aspects that might relate to certain safety aspects. It's difficult to predict at this moment. But assuming that it's safe for you to participate, you will be provided the opportunity to participate in one of our clinical studies. It is never your requirement to do so. So nothing's automatic. You're not required, but there will be that opportunity in all but an unpredictable handful of patients who, for some reason, participate. But I expect that every patient who wants to will be able to participate.
Okay, that's wonderful. We have a comment from the audience, and that comment was, "Thank you so much for considering more progressed patients in this trial. I just want to share how grateful I am as a part of the 2I community that MO Bio is being so inclusive." And that's from one of the patients that they wanted to share that. And then we have a couple more questions. Are these trials placebo controlled? And if so, will the placebo recipients have the opportunity to get the drug at the end of the trial?
Yeah, so we're still working out the exact study design. In the event that we do pursue a placebo-controlled design, so that means that some of the patients would receive a dummy drug while the others receive the drug. Regardless, even in that situation, all patients who participate in the study would be given the opportunity to enroll in what's called a long-term extension study that would be open label, and then all patients would have the ability to receive the drug on an ongoing basis. Assuming that this trial was successful and that we did ultimately gain regulatory approval to make this available to the broader patient community, that would still take additional time. So even in the event that you participated and you were in the placebo arm, you would still have the opportunity to get BBP-418 well before anyone who did not participate in the clinical study.
Okay. And then what is the process to get the drug approved in Europe?
So Europe has additional complexity. We are planning on conducting our clinical trials both in the United States and in Europe. We are expecting to pursue regulatory approval concurrently in the United States as well as in Europe and other countries. That being said, there's obviously additional national and national boards and national authorities that need to be involved and engaged and need to provide their own level of approval for a product that's actually available in those countries. So we're going to pursue it as fast as we can. We're going to work with our European regulatory colleagues and health technology assessors as aggressively and appropriately as we can. We have the full agenda that everyone in Europe does to make this product available as soon as possible. But it may take additional time because of the processes in place.
Okay, if the study site that's near me is not yet active, when can someone expect that they will start enrolling patients in the lead-in?
It's a great question. You know, our goal is to get all of our study sites open as quickly as possible. You have a couple of different options. You can email or call Brittany Holmberg, who's your overall project manager. She'll maintain your name in the queue, and she has the best sense about when that study site might be open. She would push your name to that site when it got close. If for whatever reason that seems like that's quite delayed, then we're also happy to redirect you to the next available study site. And there's travel that's supported as part of this, and you can do some of the visits remotely. So all of that's designed to be able to allow you to participate.
That's wonderful.
My one plug to that is is just to reinforce there's options. If you want to participate, reach out. And I'm sure that they can try to accommodate you in some way that'll work.
Okay, that's wonderful. And then we had some questions about study sites in Europe and in the United Kingdom. Where can somebody find out if a country is on the list? Is that on the website? Where could they go for that?
So at this point, the lead-in study, we anticipate one site. That's Dr. Vissing in Copenhagen. Our expectation is that we will probably have at least one, probably two additional sites for a pivotal study. The exact location of those sites and where they are in specific countries is still something that's very much up in the air and to be determined. I would say, assuming that we do have the ability to travel internationally within the European Union, broadly defined, that we will be supporting travel as well. So I would not look at not having a site within your country as an obstacle, provided that obviously the COVID situation allows travel across borders.
Okay, that's a very, very good answer. I'm sure a lot of people in Europe would be interested in participating. So the current site is in Copenhagen, but then there's going to be some additional sites opening up in Europe. Okay, that's good. And then last question is, will this treatment be administered orally, by IV, or injection?
That's probably a good first question. So this is an oral product. So it's going to be administered in what's called a sachet. So it's a little pouch with a powder that you'll pour under water. It'll dissolve and you basically drink it. So not basically, you drink it. So it should be something that's pretty easy and innocuous as far as the administration.
Okay, that's
no
that's,
IVs, no
IVs, no shots.
No shots, no.
That's great. Boy, everybody's wanting a medication that's just like that, just a powder. That's all, Well, I want to say thank you so much, gentlemen, for being with us today. And I want to thank you for all that you're doing for the Limb-Girdle 2I community. We're so grateful and thankful to you. You're really changing lives and making a huge difference in our lives. And so if no one's thanked you in the last week, then I just say thank you so much from the bottom of my heart for what you're doing to help us. And thank you. So everyone, we're going to make sure that all the information is listed on our YouTube platform and channel so that you'll know how to contact Brittany so that you can find out about site locations for these clinical trials. And if you have any additional questions, you'll also see that there's information in the description of this video on the YouTube channel, and then you can contact them on bio directly as well. So thank you so much. And remember, if you want to join the LGMD Patient Network with the Speak Foundation, you'll be the first to find out about all of these educational webinars that we provide. And you'll also receive a free copy of the new Limb-Girdle News magazine that's coming out in late 2020. That'll be an exciting magazine with all emerging news for Limb-Girdle and any new study that's being coming around for the Limb-Girdle community will be in that magazine. It'll be a great resource for you. So if you want that, you can go to the speakfoundation.com and sign up for our LGMD Patient Network there. So thank you so much, everyone, for being with us today, and have a great day. Thank you.
Thank you very much.
Thank you.